Hello everyone and welcome to this NDPI webinar on molecules dedicated to new trends on flavonoids in human health. Thank you very much for joining us today. This webinar is based on one of the special issues from our molecules journal. So if you are interested in submitting your paper to this special issue, all the information is available on the webinar webpage. This webinar will be chaired by Dr. Marisa Freitas, and will contain the excellent talks of Professor Eliana Souto, Dr. Daniela Ribeiro, and Professor Gary Williamson. We will also have a part for questions moderated by today's chair after each talk. So if you have any question, please feel free to post them in the QA button that you will find at the bottom of your screen. I would like also to inform everyone that this webinar is being recorded and that the recording will be posted on the conference webpage as soon as possible. We also have a YouTube live streaming today, and I'll be sharing the YouTube link through the chat in a moment. Finally, I want to present our chair. Dr. Marisa Freitas completed her PhD in pharmaceutical sciences in 2010 at University of Porto in Portugal. Currently, she is an assistant researcher at the Faculty of Pharmacy of the University of Porto, working at the Laboratory of Applied Chemistry. With over 78 publications in international journals and over 2,500 citations, he has participated in several projects funded by national and international funds. She also collaborates as a reviewer in more than 20 journals and participates on the editorial board for 4 journals, including Molecules. Thank you, Marisa, for accepting to chair this webinar. The floor is yours. Everyone enjoy.
Thank you. Hello, everyone. It is my pleasure to be here as a chair of this webinar entitled New Trends of Flavonoids in Human Health. First of all, I would like to thank to the honorable 3 speakers the prompt acceptance of the invitation to participate in this webinar, sharing their valuable results with us. Ever since ancient times, in search for rescue for their disease, the people looked for drugs in nature. Medicinal plants have been transformed into one of the oldest sciences in countries such as China, Greece, Egypt, and India. In fact, all of us, sometimes even unconsciously, use plants to treat diseases. Who has never had tea to relieve a sore throat or a stomach problem? The history of research on flavonoids goes back several decades. In 1930, a new substance was isolated from lemon juice by the Nobel laureate Albert Gheorghi, who also discovered vitamin C. At that time, it was believed to be a member of a new class of vitamins and was referred as vitamin P. Later, it became clear that this substance was a flavonoid, namely rutin. Until now, at least 6,000 different types of flavonoids have been identified. Flavonoids are an important class of natural products. They belong to a class of plant secondary metabolites, having a polyphenolic structure widely found in fruits and vegetables, contributing to their colorful pigments. The flavonoids compounds possess 15 carbon atoms and 2 benzene rings, joined by a linear 3-carbon chain, constituting one of the most characteristic classes of compounds in higher plants. The flavonoids can be divided into several subgroups classified according to the substitution patterns of C ring, and both the oxidation of the heterocyclic ring and the position of B ring are also important into their classification. It seems that a diet rich in flavonoids may be beneficial for human health. Emerging evidence from epidemiological and controlled trials support protective effects of foods and dietary supplements rich in flavonoids against chronic diseases as cardiovascular diseases, among others. However, the biological effects of flavonoids depend not only on their content in food, but also in their bioavailability that is influenced by the source type, consumers' characteristics such as gender and age, and also differences in mechanisms of absorption and biotransformation of different compounds. Therefore, the main goal of this webinar is to share experience and promote discussion on novel challenges and outlooks concerning the new trends of flavonoids in human health. For that purpose, we will have 3 speakers with a broad experience under this theme. And looking at the program, we will have about 20 minutes for each presentation. And at the end of each presentation, we will open the session for the questions. So if you have any question, please post it in the QA box. So it is my pleasure to introduce you our first speaker, Professor Eliana Souto. Professor Eliana is affiliated with Faculty of Pharmacy of University of Coimbra and with the Center of Biological Engineer of Minho, both in Portugal. She has 7 patents and more than 600 scientific publications in the field of design, development, and characterization of new drug delivery systems. Professor Eliana, thank you again, and the floor is yours.
Thank you very much, Dr. Marisa Freitas. Thank you very much for the nice introduction. And thank you also for having me here today in this webinar series of molecules. I will try to now share the screen. So let's get started after putting my voice in the right tone. So I've been proposed to talk about the use of nanoparticles for the loading and for the delivery of flavonoids. And within this topic, I would like to drive your attention to some examples or some studies that support the concept that when loading an active pharmaceutical ingredient into nanoparticles or into nanomaterials within a broad concept, so these particles or these materials may enhance their bioavailability, some may enhance their bioactivity. Bioavailability, obviously, in vivo, if that shall be the aim. And I'm going to start by showing you that from a quick search I've done on the Web of Science last Friday or Saturday, I don't recall it, using these 2 terms, flavonoids and nanoparticles. Most of the works available have been published over the last 5 years. However, the topic of loading flavonoids into nanoparticles is no longer a new topic. Actually, when we pick up a new active pharmaceutical ingredient to load it into nanoparticles for any pharmaceutical application or any technological, mostly for any pharmaceutical application. So the study design that we will put into practice is actually to figure out how much of the active pharmaceutical ingredient will be loaded into the selected system. And ultimately, we want to figure out exactly and in a quantitative manner where these materials, these nanoparticles, are in the body when administered in vivo, obviously. When are they there? How much of these materials, how much of the drug is in a given tissue, organ or cell? And what is the concentration curve over time of the drug that is being released from the selected drug delivery system? So most of the knowledge that we create is actually incremental, as we are continuously testing these systems ex vivo, in vivo, in a quantitative and time-dependent fashion. One of the drugs that we have been working with is the classical epigallocatkin galette, which comprises, as you know, about 70% of the composition of the T catechins. The drug is very popular for the recognized human beneficial effects on human health. And because of its antioxidant properties, EGCG has been proposed for the treatment of diseases of the posterior segment of the eye. Posterior segment of the eye is the most tricky section of the eye anatomy to reach. But before reaching the posterior segment of the eye for the treatment of a range of ocular diseases, the challenge here is actually to stabilize the epigallocatechin gallette, because EGCG is actually unstable in solution, so it is sensitive to many, to several experimental conditions. It degrades via oxidation, epimerization processes, which obviously will compromise its use in clinical settings. So to reach the posterior segment of the eye, which is this section here, as I said, the most tricky one to reach, ideally it should be reached by any non-invasive approach, because in this way the treatment will avoid any discomfort or any pain to the patient. So that the drug has to overcome a great deal of barriers. Besides, a non-invasive drug delivery system should provide targeted delivery by avoiding, first of all, large doses of the drug, and in this way it should also reduce the toxicity encountered by the conventional treatment approaches. So the requirements, besides the prerequisite of being biodegradable, biocompatible, it should exhibit mucoadhesion, high solubility and permeability. So that the delivery system should also be stimuli responsive and should provide a modified release profile. And because of the solid core, as the name recalls, the solid core of solid lipid nanoparticles, these SLN, I'm going to call it from now on SLN, they seem to combine all these features for a modified drug delivery. And besides, SLN can be produced with a positive surface charge to improve mucoadhesion onto the ocular surface, because ocular surface is negatively charged. So we have successfully developed cationic SLN in the presence of ascorbic acid as the antioxidant. So the idea was to dual load the dual loading of cationic SLN with EGCG and ascorbic acid. And we have produced two types of lipid nanoparticles, of cationic lipid nanoparticles. Let's say one with CETAR and another batch with DIDA. So these are two types, distinct cationic lipids. And we have checked their transcorneal and transscleral permeation profiles by measuring the cumulative percentage amount of drug over time in each of the tissues. And the results of the EGCG transcorneal and transscleral permeation from the lipid nanoparticles, they demonstrate that SLN, that our particles actually deliver the flavonoid at a constant rate and with enhanced accumulation into both tissues, into corneal and scleral. Obviously, the profile will depend on the type of cationic lipid. So actually, to use cationic lipids, cationic nanoparticles, in this case cationic SLN, as drug delivery system, importantly is to ensure that both the irritation and tolerability tests, they result in favorable outcomes. And actually our particles, they showed in fact no signs of vascular response after treating the HETCAM with the cationic nanoparticles, both batches here. And no signs of ocular inflammation, congestion, swelling or lacrimation in the DRAISE test using the rabbit eye test with a mean total score of 0. The corneas were observed to be crystal clear with intact ocular surface. So no damage was observed on the cornea, iris, and conjunctiva at all time points. To increase the ocular retention time and the mucoadhesion by these kind of electrostatic interaction or attraction, cationic nanoparticles are actually very appealing, but when using cationic lipids to obtain these positively charged particles, a significant decrease in the cell viability may be seen depending on the concentration of the cationic lipid. And the issue raised here is actually the risk of cyto and genotoxicity. And we have reported that the impact on the viability of retinoblastoma cell line can be controlled by limiting the content of the cationic lipid, in this case CETAP, up to 0.5% of SLN formulation. We need to bear in mind that the toxicity depends very much on the type of alkyl chain we are using as a cationic lipid. And also depends very much on the concentration of lipid particles we put on the cells, because the cells, they are very sensitive both to the concentration of the lipid and also to the size of the particles. In one of our previous studies, we have also demonstrated that amphiphilic and cationic lipids, they are actually not toxic at the therapeutic concentrations, because we use concentrations of lipids that are tolerated by cells, and we have seen that in most of the cases the genotoxic effects result from the concentrations of particles that already show cytotoxicity. So besides the cytotoxic trials we have to run with our drug itself alone, we have obviously to combine the drug with the nanoparticles and run void and empty and loaded particles. As EGCG is a candidate for the treatment of Alzheimer's disease, the very same concept of dual loading EGCG and ascorbic acid, as successfully done with our cationic SLN. So the very same concept we have applied or use it to load PLGA nanoparticles surface tailored with polyethylene glycol. And their effectiveness was evaluated in a double transgenic mice model as a new strategy for the treatment of Alzheimer's disease. And our polyethylene glycol PLGA nanoparticles, they were prepared by double emulsion method, optimized by a 2 to 3 factorial design approach, as it is done in each and every new formulation that we develop in the lab. And the selection of the best formulation is usually based on the relationship between the highest encapsulation efficiency, the highest loading capacity as possible for that specific compound, in our case EGCG, and with the lowest particle size, and more importantly, with the lowest polydispersity index as possible. Neuroinflammation following by accumulation of the amyloid beta plaques plays a crucial role in the pathogenesis of this disease, of neurodegenerative diseases as Alzheimer's disease, and the treatment with the free EGCG did not show any significant reduction in either amyloid beta plaques peptide accumulation, but the loading of the EGCG, the dual loading into our polymeric nanoparticles, they were more effective in reducing the neuroinflammation and the pathological accumulation of the beta plaques in mice. The behavior of transgenic mice was assessed following the EGCG treatment for a period of 3 months with the daily treatments of comparable doses of EGCG, and the data indicate that the dual loading offered a significant treatment advantage over the EGCG. So all in all, the double-loaded PLGA nanoparticles increase the spatial memory and learning capacity of the mice. They reduced neuroinflammation. They reduced the accumulation of the amyloid beta plaque burden, and also the cortical levels of soluble and insoluble amyloid beta plaques, amyloid beta in this case, and more importantly, and very important, an increase in the synapses expression, achieving similar values as the healthy individuals. Another very popular type of biodegradable delivery systems are carbon nanotubes. So these materials are cylindrical molecules that consist of rolled up sheets of single layer carbon atoms, and they can either be single-walled with a diameter of less than 1 nanometer, or multi-walled consisting of several concentric interlinked nanotubes with diameters reaching more than 100 nanometers. So their length can reach several micrometers or even millimeters. And we have used carbon nanotubes to functionalize them with naringerin. Naringerin is a flavonoid, a flavonone obtained from several citrus fruits, and exhibits a wide range of pharmacological effects. And we wanted, and actually we patented the technology, we wanted to check if the functionalization has some effect as anti-cancer approach, especially for lung. Carbon nanotubes, they commonly show a smooth surface with few defects on the side walls. Their functionalization with naringerin promoted an increase in the diameter of the samples, confirming the successful functionalization. So it is actually easy to understand that theoretically the high surface area of carbon nanotubes allows the attachment of a large number of molecules, making it as a potential drug carrier. And in this way, they may also, so carbon nanotubes, they may also be useful for the controlled release of the attached drugs. A simple release test was run using 2 distinct pH value medium to simulate the biological fluids and the tumor periphery. To evaluate the release rate of naringerin from nanotubes after acidic treatment and subject to oxidative environment. And we see that the release of flavonoid of naringerin was strongly dependent on the pH, but regardless of the pH conditions, both samples, they show they show the best fit with the Cos-Myers-Pepper's mathematical model. Estimating, so we can estimate from the square R values that the flavonoid release can happen maybe because of diffusion and relaxation of the linked compound on the surface of the carbon nanotubes. Naringenin has indeed been reported to induce apoptosis and to decrease the viability of several cancer cell lines, including the lung cancer. And concerning the cytocompatibility or cytotoxicity, we've tested in human fibroblasts, and 3 naringenin show the highest cytotoxicity with increasing concentrations, of course, while the functionalized carbon nanotubes were less toxic than the naringenin alone, indicating that functionalization promoted the decrease in the drug cytotoxicity, so leading to possible reduction, also potential reduction of these side effects. Both functionalized carbon nanotubes and naringenin alone significantly inhibited the viability of malignant lung cells. And the cytotoxicity inhibitory concentration results indicate that the functionalization exhibits a higher cytotoxicity, again, against or in comparison to the free naringenin, meaning that naringenin has indeed an enhanced anti-cancer effect when functionalizing the acid-treated carbon nanotubes. Before finishing, one short note on the simplest way to obtain drug delivery systems from lipophilic compounds, which is the easiest way to produce delivery systems with oils and whatever. So it is the production of simple oil in water emulsions. And this is a very simple work in which we have used neem oil. Neem oil is rich in quercetin. We use neem oil to obtain nanoemulsions, again, optimized by factorial design approach and testing different stirring methods. Neem oil or a neem tree is a fast-growing and evergreen tree that has been used in folk medicine since the early times of human existence. So the composition of neem oil in fatty acids is very rich, and neem oil has been proposed as a pesticide besides its potential use in pharmaceutical industry, given its anti-inflammatory, anti-tumor, antifungal activities. And very briefly, the antifungal activity of our developed nanoemulsions was tested against saprolignia as the in vitro model, evaluating the effect of different concentrations of nanoemulsions on the growth of the mold. And all groups treated with a nanoemulsion or even with a surfactant alone, surfactant solution, they showed a lower mycelium growth compared to the control group. The effects of the administration of the nanoemulsion on the antiprotozoal activity at different concentrations are shown here, and we see the highest tested concentration exhibited 100% of death of the parasites. Our study actually demonstrates that the nanoemulsion content in neem oil is indeed very effective against multifilus. I'm going to call it IgC, which is easiest. So by epifluorescence microscopy, we see the live parasite on the left-hand side of this slide, if not treated with the emulsion, and the cellular membrane damage and parasite death when treated with the highest tested concentration. And that's it. Thank you very much. And I end up with a great thanks to my international network for the possibility of a students exchange among this group. And back to Marisa.
Thank you, Professor Eliana, for your amazing presentation with a lot of interesting results. So we will open the session for questions. I will kindly ask Professor Eliana to check the QA box, and we have 5-7 minutes to answer some questions. So please select some of them and reply. Do you see, Professor Eliana, the questions?
Yes, I do see, yeah.
Okay, perfect.
Well, one of the first questions is here, as far as I understand, the metabolic pathway of EGCG nanoparticles. Well, in principle, when we think about the lipid nanoparticles, the lipid nanoparticles will follow the same metabolic pathways as the lipids, if they are given orally, as the lipids that figure in our food. So they will suffer all the metabolic pathway, so production of mast cells, and we may take advantage of the fact that lipids, they act as absorption enhancers. And in this way, any drug that is loaded into those particles, they will, in principle, be will offer the opportunity to increase the bioavailability. With respect to the run assays of naringerin on human cells, we haven't done it yet. We have only patented the technology, the functionalization approach. The next step would be running in vitro and in vivo experiments. There is one question continuing the topic of carbon nanotubes, how they are functionalized. Well, actually, we treat the carbon nanotubes. They are purchased, they are commercially available, and what we have to do before combining them with the drug, in this case with the flavonoid, we treat the carbon nanotubes with acids, usually with nitric acid and with sulfuric acid, a mixture of those, and to expose the functionalization groups, so that the interaction with the drug can happen. So how much of flavonoid is linked to the nanotubes? Well, the ratio between the carbon nanotube and the flavonoids is 1:1. So in principle, we would have less than 50%, less than 50% of drug combined, attached, in principle, also absorbed onto the surface of carbon nanotubes. Carbon nanotubes, they have different possibilities to different spots where they can be combined with the drug, but the easiest way is the adsorption. continuing the topic of carbon nanotubes, the possible administration routes, we haven't done that yet, but from the literature, they have been tested in animal models by intravenous administration, by a tail vein injection. So we will, in principle, use the very same approach. And
the last one, please.
Sorry.
That's okay.
Okay, one last one. The EGCG permeability in the eye tissues. This is a very interesting topic for me because this is my main research topic. Actually, it is indeed dependent on the type of cationic nanoparticles. If you use a single chain cationic nanoparticles, lipid nanoparticles, they will suffer a higher permeation rather than retention in both of the tissues. But if you use a double chain cationic nanoparticles, it will happen the opposite. So it will be a higher drug will be retained on those tissues and sclera, and from once retained in those tissues, they will provide a sustained release profile. It is a very interesting topic that requires further attention. Thank you very much.
Thank you, Professor Eliana. So let's move on for our second speaker. I will ask Professor Eliana to stop sharing her screen. Yes. So our second speaker is Professor Daniela Ribeiro. Professor Daniela Ribeiro is an assistant professor in the University of Açores in Portugal. She published 5 book chapters, 48 papers, and presented almost 100 poster and oral communications. She has been working on the antioxidant and anti-inflammatory activity of flavonoids and their derivatives. Professor Daniela, thank you, and you can start when you want.
Thank you, Dr. Marisa, for your kind presentation. Good morning or good afternoon. First of all, I would like to thank the organizing committee of this webinar for this opportunity to present the work we have been developing entitled Flavonoids and their Styryl derivatives as promising modulators of pro-oxidant and pro-inflammatory pathways. So I will start this presentation with a brief introduction to flavonoids and their derivatives, styrylchromones, and on inflammation. Then I will present the methodologies used to evaluate these compounds, pro-oxidant and pro-inflammatory, the flavonoids and styrylchromones' ability to modulate pro-oxidant and pro-inflammatory pathways. Then I will present the results obtained in an integrated form. And finally, I will present you the overall conclusions. So, polyphenols are aromatic plant metabolites, and at least 10 different classes are known, and most notably hydroxybenzoic acid derivatives, cinnamic acid derivatives, tannins, and flavonoids. As Dr. Marisa already said, flavonoids have a C6, C3, C6 aryl benzopyran core, and their derivatives or steryl derivatives, styrylchromones, have in common with them a chromone core that you see here in purple. But styrylchromones have also a styryl group attached to carbon number 2 of the chromone core. Flavonoids and 2 styrylchromones can be obtained by chemical synthesis, but they also occur in nature. So flavonoids, as Dr. Marisa has said, are distributed in nature. They are identified several subclasses, and we can find them in our daily diet, as in fruits, vegetables, and even in beverages. Two styryl chromones also occur in nature, but they are scarce. So as far as we know, only 9 2-styryl chromones are already identified. So they are mostly obtained by chemical synthesis. Two flavonoids and two styryl chromones have been attributed antioxidant effects and the modulation of intracellular signaling pathways. These effects are believed to are involved in their health-promoting properties. So flavonoid and two styryl chromones have been studied for their anti-allergic, antifungal, antiviral, antitumoral, antibacterial, and anti-inflammatory properties. In what concerns inflammation, we all know that it is a physiological response of the organism to an aggression. It is a complex and dynamic process consisting of multiple and interdependent cellular and humoral changes. As we all know also, the inflammatory response is really complex and it comprises a myriad of processes, as you can see here, that are interregulated and most important of all, they regulate each other. And they are comprised by the production of cytokines and chemokines, the activation of the complement system, the activation of transcription factors, activation and recruitment of inflammatory mediating cells, but also by the production of pro-oxidant reactive species and production of eicosanoids, namely prostaglandins. So here I'm going to compare the antioxidant and anti-inflammatory activities of flavonoids and their styryl derivatives to styryl chromones based on the work we have been developing in our research group. So for this purpose, we selected a panel of structurally related flavonoids and two styryl chromones. And you can see here, they may have none, one, or two hydroxy groups on A ring, and also none, one, or two hydroxy groups on B ring. So I will first show you the study of the modulation of reactive species production that we have done it by two different methodologies. So first of all, we can study this by the direct scavenging of reactive species. And here I show you that the first step of this methodology is based on the generation of reactive species. They may be oxygen reactive species or nitrogen reactive species. So then we incubate this reactional mixture with the compounds under study, and then we add the respective detection proofs. So these proofs may be colorimetric, chemiluminescent, or fluorescent proofs, and we select them according to the reactive species that we want to detect. Overall, if we have compounds that are good scavengers of these reactive species, we'll see at the end a decrease in the detected signal. The other methodology that we can use is the assays for the study of the modulation of human neutrophils' oxidative burst. In this case, we start to isolate the human neutrophils from human blood that are collected from volunteer blood donors. And we do this by using the density gradient centrifugation method. So at the end, we'll have the isolated neutrophils and we assess their viability. In this case, I will not present these assays, but of course that all the compounds that were studied were evaluated. We have evaluated their cytotoxicity to guarantee that they do not affect the human neutrophils. So once we have the isolated neutrophils, we activate them with PMA that will lead to the start of reactive species production. And to detect this production, we use the chemiluminescent proof luminol. Luminol is not a specific proof, so with it we can detect reactive oxygen reactive species and nitrogen reactive species. However, luminol enables us to detect these species intra and extracellularly. So if our compounds are good inhibitors of reactive species production, we will then see a decrease in the detected signal. So for the results, I will start to show you first the results for the scavenging assays and then for the oxidative burst assays. So here is a table that presents you the most active found flavonoids and 2-styrylchromones on the scavenging of reactive species. This is a simplified table because a lot of data was generated, but here you can see that the results or the IC50 values found are variable according to reactive species detected and to the compounds, of course. However, we can highlight that a significant number of them retrieved us low values of low IC50 values, and also some of them have a feature in common, that is a catechol group on B-ring, here R1 and R2. Also, we can see that 2-styrylchromones seems to be a little more active than flavonoids. When you look to the inhibition of the neutrophils oxidative burst, here are presented the most active compounds, and we can also see that some of them are similar or are the same that I have presented in the previous slide, so they are also good scavengers of the reactive species. Here we have also low IC50 values, and once more 2-styrylchromone seems to be more active than flavonoids. And also once more, we have the same feature. So we have a catechol group in common in the B-ring. Then I'm going to present you the studies for the modulation of prostaglandins production. And here we also apply two methodologies. The first one is based on the evaluation of the COX-1 and COX-2 activities. In this case, the tested compounds or the compounds under study are studied directly in contact with the isolated enzymes, the ovine COX-1 and the human recombinant COX-2. And then the prostaglandin that are produced are quantified by ELISA, and all of these are made through commercially available kits. In the second methodology, we evaluate the modulation of prostaglandin E2 production via COX-1 and COX-2 in human whole blood. In the case of the COX-1 assay, this first one you see here, we incubate the human whole blood with the tested compounds and with calcium ionophore. In the case of the COX-2 assay, we also incubate the human whole blood with the tested compounds, and we had the lipopolysaccharide that will induce COX-2 expression. So the incubation time in this methodology is a bit longer, so we can have the expression of COX-2 and the production of the prostaglandin. I would also like to highlight that the use of human whole blood retrieves us a condition much more similar to the one that we found in the physiological environment. So once we have the prostaglandin produced, we quantify it by an ELISA assay, and in this case, if our tested compounds are good inhibitors of prostaglandin production, we'll have less in our sample, and we will observe an increase in the absorbance signal. So as results, I will first present you the results obtained for the COX-1 assays and then for the COX-2 assays. So here we have the flavonoids that were the best inhibitors of prostaglandin production via COX-1. As you can see, looking to their structures, they have a low number of substituents. And those were the two Styryl chromones that were the best ones on the COX-1 assay. But also here you can see that the flavonoids are best inhibitors than the two Styryl chromones. Looking to the COX-2 assays, those were the flavonoids which display the best activities, and they all have in common a catechol group on B ring once more. However, none of the tested two Styryl chromones that we studied were active. Here, I would like to tell you that flavonoids were tested using the human whole blood, a much more complex matrix. And two Styryl chromones were tested with the isolated enzymes. So here, these results are really interesting for us. So as conclusions, we can say that chemical features that define the best compounds are common to both flavonoids and two Styryl chromones, and the catechol group on B ring seems to be essential for the overall activity. Flavonoids and two Styryl chromones were then good scavengers and good modulators of reactive species production, but interestingly, flavonoids were best inhibitors of prostaglandins production via COX-2. Overall, both flavonoids and two Styryl chromones display promising modulatory inhibitory effects of pro-oxidant and pro-inflammatory pathways, but obviously chemical assays are here needed to improve these structures, because we need these compounds to be more target specific, and this is why our research is still going on. Finally, I would like to acknowledge all the people from my research group, especially to the PhD student Mariana Lucas, to all the collaborators in this work, and obviously to the financial agencies and all of you for your kind attention. Thank you.
Thank you, Professor Daniela, for your very nice talk with interesting molecules that can be used as anti-inflammatory agents. So you can please check the Q&A box. I think that you have two questions.
Okay, so the first question, they're talking or questioning about the products of reaction between flavonoids and reactive oxygen species. We didn't study the reaction products. We just studied the scavenging capacity of flavonoids and the modulatory effects on human neutrophils. So the products, we did not study them. One more question. So here it is an interesting question, mainly about comparing I think the bioavailability of Styryl chromones and flavonoids, which is a really interesting question. So they have a similar structure. So in terms of bioavailability and metabolism, they are really similar. So we know that Styryl group attached to chromone core stabilizes the molecule of two Styryl chromones, and this is the advantage when comparing to flavonoids. But in terms of bioavailability, I believe they will be really similar, despite the fact that we have much more studies for flavonoids than for two Styryl chromones that are really less studied compounds by the scientific community. So we have one more question. So we did not detect the expression of COX-2, okay, we just evaluated its activity. It is also done in our lab, but here I did not present those assays. Those assays are interestingly now being done because we have another project. So in the future, we believe that we'll have both informations, effects on the activity and effects on the expression of the enzyme. Okay, so one more question about the cytotoxicity of the compounds. As I said in my presentation, I didn't present these results because we don't have time, but yes, we always study the cytotoxicity of our compounds. Here we study human neutrophils, isolated human neutrophils. So we need to try all the concentrations we test to see their cytotoxicity to guarantee that they do not affect the neutrophils' viability, but also because we tested whole blood, human whole blood, we also need to test the cytotoxicity of these compounds, for example, in erythrocytes and the leukocytes, the overall leukocytes. So yes, we have to guarantee that the concentrations of the compounds do not affect the viability of the cells. This is the first thing to do.
Professor Daniela, did you see any difference in the viability between flavonoids and the Styryl hormones?
It depends, because flavonoids, no, they were not toxic, and these Styryl hormones that I presented here were also not cytotoxic, and this is why they were tested and continue in our studies. However, we have now a project going on specifically with the Styryl hormones, and we are seeing some different behaviors, but we believe that it is because we have new molecules with different substitution patterns, and probably the new substituents in these molecules are contributing for the cytotoxicity that we are now finding for Styryl hormones.
Thank you so much, Professor Daniela.
Thank you.
So let's move on for our last speaker, Professor Gary Williamson. Professor Gary teaches in the School of Clinical Sciences at Monash University in Australia. Professor Gary is a highly cited author with a H index of 98 and was in 2015 included in the list of the world's most influential scientific minds published by Thomson Reuters. He has been working in polyphenol bioavailability, and more recently, he has been working in the modulatory effect of polyphenols in type 2 diabetes. Thank you, Professor Gary, for your presence in this webinar, and the floor is yours.
Thank you very much for the introduction, and thank you for the invitation to present. I'm just going to share my screen. So I'm going to talk about the effect of dietary polyphenols on insulin resistance. And this is an area where I think that polyphenols in the diet have a big impact on health. And I'm going to give you an introduction to the area, to a little bit of information on insulin resistance, and then how polyphenols might affect that, focusing on polyphenols in the diet. So the sorts of compounds that we're interested in are generally called phytochemicals, and polyphenols are a group of compounds within the phytochemical class. There's also glucosinolates, which is a minor class in brassica vegetables, and there's also carotenoids. But they all contribute in some way to the properties of the particular foods, fruit, or vegetables. So, for example, the flavonoids, which we've seen already, have this 3-ring structure, whereas phenolic acids generally have one ring with a carboxylic acid group, or they have multiple rings which are just many phenolic acids linked together, whereas the carotenoids have a much more sort of linear structure with and don't always contain aromatic groups. So the distribution in foods is quite distinctive. It tends to be that foods with a high content of polyphenols generally have a low content of carotenoids. So if we look here, we can see that blueberry, cherry, raspberry, blackberries are all high in polyphenols, whereas things like pepper, spinach, and carrot are high in carotenoids. So this means that when we're looking at the effect of different polyphenols, often people will use different types of food rich in polyphenols to look at an effect rather than the pure compound itself. So we've heard that there are many thousands of different types of flavonoids, and there's even more types of phytochemicals, which makes for a quite complex situation. But if we actually start to look at the metabolism of these compounds, then in some ways the situation starts to get more simple, and we don't have to consider quite so many compounds. So if we look at the phenolic acids, then these can be broken down into several different types of compound by the gut microbiota. So we start off with the chlorogenic acids, and this is different phenolic acids linked to quinic acid, which are all of these ones in the middle. But once the gut microbiota, or the body, starts to break down these compounds, you actually end up with a less number of different compounds, because the phenolic acid is released. And so consequently, the phenolic acid, there's only a few types of phenolic acid compared to chlorogenic acid. So the situation becomes more complicated, it becomes more simple. But once the phenolic acids have been released, then they can be metabolized further, they can be sulfated, and they can be converted into other metabolites. And so the situation becomes slightly more complicated, but there's less metabolites found in the body than there are parent compounds because of this breakdown. So I'm going to talk about the effect of these different compounds on insulin resistance, and just briefly go through what insulin resistance is. So insulin resistance is almost entirely caused by diet and lifestyle factors, and in many cases can be reversible. So you have the healthy person with a poor diet, high in energy, too many calories, low in plant foods. Person can develop over the course of years a situation called insulin resistance, which is on the way to developing type 2 diabetes. And what it means is that actually the tissues respond inadequately to insulin released from beta cells. So this process is actually in some ways reversible. If you have the right food, diet, exercise regime, then you can reduce insulin resistance and become more healthy. If not, then insulin resistance can develop into type 2 diabetes and into further diabetic complications, and both of these really need more drug treatment. So we're most interested in the situation between healthy and insulin resistant. So if we look at how people might respond to intake of sugar in different situations. So if we look at, first of all, at a lean, normal person, then if you give them an oral glucose load, so you feed them glucose and then measure glucose in the blood, then there is a peak of glucose, but it rapidly returns back down to the baseline value, which is around about 4 to 5 millimolar. In people who are obese, it increases a bit more, but soon again returns to normal. In people with insulin resistance and with diabetes, the glucose stays around for much longer in the blood and reaches a much higher level. And this can cause many different problems related to this high glucose concentration. In addition, the insulin secretion in lean normal people follows a similar curve, goes up and then goes back down again. But if you look then obese people, it increases even more. But in people with insulin resistance, it increases very much more, even more than people with diabetes, because the body continues to try to produce insulin because glucose is high, but the tissues are less responsive. So consequently, more and more insulin is released into the bloodstream. So if you look at the real-time glucose concentrations in real people, then it doesn't follow those sort of simple curves, which I just showed you in the previous slide, because those are based on laboratory experiments. If you continuously monitor blood glucose in free living individuals, then it looks a bit like this. It's really messy. But what you can see is that the range of response in healthy people is much, much lower than the range of response in patients who are insulin resistant. And this follows throughout the day. And you can see there's a lot of difference between individuals as well in this glucose fluctuations. So one of the things that's been used to treat type 2 diabetes, or at least improve the condition, is the drug called acarbose. And acarbose is a tetrasaccharide which inhibits carbohydrate digestion. So inhibits starch digestion by inhibiting amylases. It's not absorbed, but it absorbs, it inhibits amylases and also inhibits maltase and sucrase, all of which are enzymes involved in the digestion of starch, leading to the incorporation of high levels of glucose in the blood after a meal. Because acarbose is not absorbed, it's a useful way of looking at the effect of reducing this after meal or postprandial glucose. And what happens is that the incidence of diabetes is substantially reduced by administration of acarbose with food. So the question is, can polyphenols act in the same way as acarbose, reduce the amount of glucose in the blood after a meal, and therefore reduce the risk of type 2 diabetes? So let's have a look at first of all, at the epidemiological evidence for reduction in risk of type 2 diabetes by, in this case, by tea and coffee consumption. So this data is quite strong. It's based on hundreds of thousands of people from many, many different studies. And the bottom line is that coffee consumption dose-dependently reduces the risk of developing type 2 diabetes, therefore improves insulin resistance, as does tea. And this is from people who've been looking at people and looking at their habitual dietary intake to see how their risk of diabetes is affected. You can also do experiments on people. You can do human intervention studies, and you can look at the effect of giving different either compounds or flavonoid-rich foods on the different markers related to insulin resistance and type 2 diabetes. So each one of these columns in the middle shows the number of human intervention studies that have been done. So quite a lot of studies have been done on people and flavonoids and health benefits. So in this particular case, quercetin has been shown to lower blood glucose, lowered cholesterol, and lowered blood pressure, all of which are beneficial effects and tend to reduce the consequence of insulin resistance. It does also some other things at higher doses, probably greater than available from the diet, but available through supplementation. But as you can see, quercetin in the diet has a number of different effects related to loss of related to improvement in insulin resistance. Similarly, for anthocyanins, anthocyanins also have beneficial effects on biomarkers of insulin resistance. And again, the number of studies here, so large numbers of studies which have been done, human intervention studies looking at groups of people where you can see various different improvements, for example, in HOMA-IR, which is a marker of insulin resistance, anthocyanins lower type 2 diabetes risk. So a range of, again, a range of different effects on improving insulin resistance by polyphenols. So one of the mechanisms, as I mentioned before, by which polyphenols might work in this respect is the inhibition of alpha amylase. And in alpha amylase, we've shown is inhibited by several different polyphenols. In this particular case, I'm showing the data for punicalagin, which is a polyphenol from pomegranate. So what we looked at was the different mechanisms by which punicalagin might work. So this middle graph shows the inhibition of human alpha amylase is quite a strong inhibition, but inhibition of maltase and sucrase is quite weak. In this first graph, the inhibition of glucose transport is really quite weak. So the main mechanism by which punicalagin probably works is inhibition of alpha amylase during the digestion of starch. And then we also looked at how this worked in humans. So we gave bread, which is a good source of starch, to 16 volunteers, and we showed that the addition of pomegranate juice high in punicalagin reduced the glucose spike, the glucose peak concentration in humans with co-consumption. So punicalagin reduced these glucose spikes in the same way that acarbose did. So this is an acarbose-like effect of a polyphenol, and is one of the mechanisms by which polyphenols might reduce insulin resistance, since high glycemic index diets, so diets high in carbohydrates, especially high in starch and in various sugars, can increase the risk of developing insulin resistance and type 2 diabetes. So that's one mechanism, and that doesn't require absorption of the polyphenols, but polyphenols can also be absorbed and affect the systemic metabolism and directly affect the molecular pathways involved in insulin resistance. So one of the important factors that plays a role in polyphenol bioavailability is the gut microbiota. So the gut microbiota are in the colon. There's many bacteria in the colon. There's a lot less in the small intestine and the stomach, but the colon is very important. And the colon is where, for example, dietary fiber is converted into butyrate, and butyrate is a beneficial component of the diet. So what does the colonic microbiota, what do they do to polyphenols? So this is an experiment that we did quite some time ago. This was where we took some procyanidin. So this is procyanidin B2, and it consists of 2 flavonoids linked together by direct carbon-carbon linkage. And we radio-labeled the molecule, and then we gave it to rats. So what we found was actually that the bioavailability based on the radioactivity of procyanidin B2 was more than 80%. However, if you give the procyanidin on its own, not radio-labeled, to humans, then the amount in plasma is extremely low. So what this tells us is that the parent compound, the procyanidin, is actually very non-bioavailable. Hardly any is absorbed, but actually most of it is absorbed after catabolism and metabolism by the gut microbiota. So consequently, what is being formed from the procyanidins by this process? I'm not going to go into that particularly today, but I am going to go into the case of what is formed from chlorogenic acids from coffee. So this is a profile in the blood of people given coffee, just one cup of coffee, and then we can measure the different phenolic acids in the blood. So this is FAS, is ferulic acid sulfate. This appears fairly quickly and then sort of levels off. This is a dimethoxycinnamic acid, which appears very quickly and at very high concentration. Eventually levels off, but as you can see, it stays around for quite some time. So this is then dihydrofluoric acid and dihydrofluoric acid sulfate, both of which are microbial metabolites and produced by the gut microbiota. So the first two metabolites are produced in the small intestine and are a result of human metabolism, whereas these two metabolites, dihydrofluoric acid and dihydrofluoric acid sulfate, are produced by the gut microbiota. So consequently, the gut microbiota give us an extra boost, if you like, of different polyphenol metabolites, which we can then measure in the blood. The parent compound, which is, in this particular case, is beryl-quinic acid, which is actually present in coffee, never gets up to a very high level and is only just detectable. So a summary of this part is that the gut microbiota convert large unabsorbed molecules such as flavonoids into smaller catabolites, which can be then absorbed. These catabolites may be present at high concentration in the blood and exhibit activities which are different to those parent compounds. And the gut microbiota also play a big part in the difference between people. So then if we try and put all of this together, we put together what I've tried to tell you about insulin resistance and about the different polyphenols. So this is what's happening in skeletal muscle cells. This is what's happening after consumption of carbohydrates. So the carbohydrates or sugars are converted into glucose. Glucose stimulates beta cells to make insulin. Insulin then goes in the blood and goes through the insulin receptor into skeletal muscle cells, causing a whole range of different cell signaling processes, which ultimately lead to the translocation of GLUT4, which is a glucose transporter, to the membrane, which then takes up the glucose. It's a very complicated process for something that seems to be quite simple, but this is actually the very key, the very core of insulin resistance, because this is the insulin response. And if this gets messed up metabolically, then you will develop insulin resistance. So this is a really key pathway. Several other factors in the diet are involved. Saturated fatty acids can make the situation worse by inhibiting some of these pathways. And also very high levels of glucose can generate things like superoxide in the cells, which can react with nitric oxide and stop some of these processes from occurring. So consequently, after a meal, insulin is secreted by beta cells, which then stimulates glucose uptake into tissues, especially the muscle. And this leads to this rapid clearance of glucose from the blood in people who are metabolically competent, i.e. not insulin resistant. And this is why the peak of glucose goes up and goes rapidly down in people who are healthy. But in the case of insulin resistance, that doesn't happen so effectively, and you get a dysfunctional response. Now, as I showed you before, polyphenols in the diet in human studies can very clearly improve the insulin resistance in many different people. I showed you the example of both quercetin and of different anthocyanins. So you can see in human studies that is the case. So let's have a look a little bit about the mechanism by which these things are occurring. So what we did is we took these human muscle cells, and these myoblasts in cell culture differentiate into cells which look very much like muscle cells in culture. And we can then treat them with things like insulin, see that we get glucose uptake, and we can look at various muscle markers to see that they really are muscle cells. So we then looked at the effect of different polyphenol metabolites on the uptake of glucose into skeletal muscle cells. So we're effectively here looking at whether different polyphenol metabolites can actually act in an insulin-like manner on muscle cells, which must be a benefit if they're present after a meal. So here we have ferulic acid sulfate, which is one of the compounds in coffee, which is absorbed fairly quickly after consumption of coffee. Here we have dihydroferulic acid sulfate, which is a microbial metabolite of coffee. And we have also the dihydroferulic acid with no sulfate. So these two compounds increased the amount of uptake of glucose. It's actually 2-deoxyglucose, but it's a surrogate for glucose into the muscle cells. In addition, another compound, isovanillic acid sulfate, also dose-dependently increased the amount of glucose taken up into muscle cells. So these two compounds here are catabolites from coffee. The isovanillic acid sulfate is a catabolite from berry anthocyanins. So both of these are products of polyphenols, but not products, but not compounds which are present in the foods. They are actually products of metabolism once they've been consumed by people. So how does this work? So many of these different steps can be involved. What we found during these particular experiments was that in particular the kinase here, PI3 kinase, is important in this particular step. So we can say that the isovanillic acid and the ferulic acid sulfate and dihydroferulic acid sulfate are acting in these steps. I've indicated in yellow the steps where polyphenols can particularly interact, but in this particular instance, the PI3 kinase is probably one of the most important steps. So as a summary, I can say that dietary phenolics may affect postprandial glycemic response, so affect the glucose in the blood after a meal and blunt those glucose spikes, which is then an improvement in the potential development of insulin resistance. After microbial action in the gut by bacteria, potentially bioactive phenolics are released and absorbed in the colon, and this can boost the properties of the original molecules which are present in the food. Those microbial products can interact with other tissues, and I've shown you today muscle cells, and stimulate the uptake of glucose, and therefore enhance the insulin action and maybe get around insulin resistance. And this is supported by human studies, human intervention studies. In particular, isovanillic acid sulfate stimulated glucose transport in muscle cells. And as I said, the intervention studies show that some polyphenols protect against development of insulin resistance. So I tried to show you, link together the mechanistic studies and the human intervention studies with metabolism to show you how this sort of complex system can lead to the improvement of insulin resistance by polyphenols and therefore lead to improvements in metabolic health. So I'd like to end by acknowledging the various different people who've contributed to this work, to the different projects. And I'd also like to thank everybody for their kind attention. Thank you very much.
Thank you, Professor Gary, for the excellent and clear presentation. We have some questions in the QA box. So I kindly ask you to select some of them. We have 8 questions until now.
Molecular weight range, flavonoids around about 300. Phenolic acid smaller, about 180. Well, is there any effectiveness of tea on benefit for human health? There's probably thousands of papers actually talking about the effect of tea on human health. So yes, there are, but there's obviously not time to talk about all of those in the presentation. The bioavailability, when you label with when you label a polyphenol with radio labeled C14, you don't change the chemistry. You only change the ability to detect the molecule physically using scintillation counting. So the point about this is not the fact that it's C14, it's the fact that you can improve the amount of compound, original compound that gets absorbed by the gut microbiota, because they convert unabsorbed polyphenols into smaller phenolics, which are then absorbed. So it's not really to do with the fact that there's a label. That's purely a way of seeing what's happening. Prebiotics, yeah, I mean, they are in some ways prebiotics, because very few of the coffee phenolics would be absorbed if it wasn't for the gut microbiota. So chlorogenic acids, as I showed you, like the FQA, 5-FQA, very poorly absorbed. But once the gut microbiota act on them, then they are much better absorbed. So in some ways, they are acting as prebiotics. So do I think that there's any advantage in using polyphenols over carbos? The problem with the carbos is that it's a bit too strong. So you can actually get substantial amounts of carbohydrate going through the small intestine and into the colon, which can lead to problems with over-fermentation by the bacteria and discomfort, in the same way that people who take lactose, when they're lactose intolerant, it's a similar sort of situation. So in some ways, polyphenols could have an advantage over acarbos because they're not so powerful. But then on the other hand, if they're not so powerful, maybe they don't have an effect at all. So it's somewhere a balance in the middle. So all of those studies about this, so at what level of polyphenols can reverse insulin resistance, those studies that I showed you with by human intervention studies are actually using a hugely different range of different compounds, different foods, etc., etc. So it's very difficult to say exactly what concentrations are needed. We tend to think normally in terms of dietary concentrations that are needed, rather than high supplemented concentrations. But sometimes you need somewhere some sort of concentration in between, but it's quite difficult to generalize. I don't think that really polyphenols cause many side effects. They generally have a very, very low toxicity. The most bioactive one, quercetin, does have, I suppose, if you had a really, really high concentration, could lead to some toxic effects. But I think that at really achievable concentrations, it's not going to happen. And then there's another one, why polyphenols are not absorbed in the upper GI tract. Some of them are. The ones I chose to show you today are not. But if you take something like quercetin, that's absorbed in the upper GI tract. It depends on what attached sugars there are. So an aglycone with no attached sugars will generally be absorbed in the upper GI tract. But if you add a sugar or a quinic acid, then that needs to be removed before the polyphenol is absorbed in the body, in the upper GI tract. And actually that's what the microbes in the colon, that's what they do. They remove the sugar from polyphenols, they remove the quinic acid from chlorogenic acids, and they release the phenolic or the flavonoid. Sometimes they break that down a bit further, but then either that molecule or the breakdown products are then absorbed in the colon. So they can be absorbed in the small intestine, but generally it depends on the presence of the chemically attached sugar.
Professor Gary, do you have any comments about the nano-encapsulation of polyphenols to be used as in the treatment of diabetes?
Well, it depends on the purpose of the nano-encapsulation. I mean, if it's to improve the stability, then there should be a beneficial effect. If you get an effect, if you want an effect in the small intestine on the digestive processes, then that will occur in the gut lumen. So if the encapsulation can improve the solubility or delivery in the small intestine, that could have an effect. On the other hand, if the nano-encapsulation improves the delivery to the colon, then that could increase the microbial breakdown products which are produced. So potentially there could be, at least in theory, some beneficial effect of doing that. But that's not something that I've looked into myself.
Okay, thank you. I don't know if we have any question from the audience. I think that we don't have more questions. So thank you again, Professor Gary. So before we finish this webinar, I would like to thank again to our speakers, to the audience for this valuable discussion, and also for the MDPI for the support in organizing this webinar. I want to call your attention for the special issue, New Trends in Polyphenols and Human Health. Feel free to contact us if you are interested in publish your work in this special issue. So thank you all for your participation. I hope that you enjoyed this webinar and hope to see you soon in my beautiful city, Porto, in Portugal. Thank you very much. Anna, I don't know if you want to say something.
No, I just thank you all for your wonderful talks, for all the discussion, for all the questions from the audience. Have a nice day and take care.
Thank you. Bye-bye. Thank you.
Bye.
Thank you. Bye-bye.